Hello students, today I am going to tell you about a mutation, different type of mutation, right? And then after we will finish this chapter, that is our molecular basis of genetic inheritance. The first mutation already we know through our NCERT that mutation means what? Sudden changes, right? Which are inheritable in new generations, right? Okay, mutation means mutation occur. Mutation can occur in both germinal or somatic cells. Germinal cell means if it is present in germinal cell, it can be inherited in generation after generation. But if it is it occurs into the somatic cells, so it is non-inheritable because somatic cell is not involved in the sexual reproduction, right? In the formation of gametes, so it is not inherited in the new generations. Okay. So now first we start from mutation. Mutation, sudden changes in DNA, right? DNA strand, that is called mutation. Okay, which is inherited. Okay, mutation means what? Simply we can say sudden changes which inherit in new generation, or we can say it can transfer this generation after generation. That is called mutation. How many types of mutation? Because here it is not very important. We will read this mutation, how a mutation occurs in the next chapter, right? How many types of generation? We here read only types of generation. Types of mutation. Types of mutation. How many types of mutations are there? Right? Before going to types of mutation, first who used this term mutation? So first this term of mutation used by, this term is used by Hugo de Vries. Right? Hugo de Vries. And he observed this mutation in evening primrose. Observed in evening primrose. Evening primrose. That is important, right? Because in the Mendel's law, when we read when we read the Mendel's law, at that time I will also tell you Hugo de Vries, that is his experiment plant is evening primrose, right? Evening primrose, sometimes scientific name of evening primrose is also given. That is what happened. What is the scientific name of this evening primrose? It is Oenothera, right? It is called Oenothera lamarckiana. Oeno, that is O-E-N-O, Thera lamarckiana. This one is the scientific name of this evening primrose. It is the botanical name. Right, of this evening primrose. Okay, that we can say here who is the father of mutation, means it is Hugo de Vries, right? Morgan also observed some mutation in Drosophila. Morgan, right? Another is there, that is Muller. Morgan, Muller, they all are related with one is also Straight, right? These are the scientists who observed the mutation in their experiments, right? Beadle, Tatum, one more important is there, Beadle and Tatum. And Tatum, you know these two scientists, right? They induced they induced mutation in neurospora. You know neurospora, it is a experiment plant in neurospora, right? By using ultraviolet rays. By using ultraviolet rays or X rays, right? They induce mutation. Normal mutation means that is natural mutation. It can grow in the minimum culture medium, means a medium in which minimum level of mutation is present, it can grow. But when a mutant is induced neurospora, mutant neurospora, it cannot survive in minimum nutrient culture, right? As for example, here we can say here wild, because it is a natural wild neurospora, right? It is converted into, means it after mutation with the using of ultraviolet rays, right? Ultraviolet rays. What happens? This becomes mutant neurospora, right? So this becomes mutant neurospora. What is the difference between these two? I tell you, right? This one is called prototrop and this one is called auxotrop, right? Prototrop. Primitive. Proto means what? Primitive. Trop. Autotrop. Heterotrop. You know that this trop comes from this, and proto means primitive, and this one is called auxotrop. What is the importance of this? This means he understands that in mutation what happens. Wiedel-Tatum, they understand that during the mutation what happens. One mutant means one metabolic block, right? So they observed that this neurospora, it can survive or growth, it can grow in minimum nutrients, right? Minimum nutrient culture, right? But this one cannot survive. Why cannot survive? Because here after mutant what happens? This is actually it is a deletion type of mutation is there. So the part of DNA which is deleted, right? That means this part which is deleted, it is responsible for the formation of specific enzymes. Now these enzymes are not formed, and if due to the absence of these enzymes, what happens? It cannot survive or it cannot create or forms specific nutrients in the body. This one prototrop means it can form all types of nutrients inside the body. So it can survive here, but it cannot form all the nutrients inside the body or the enzymes inside the body because due to the deletion of some DNA. That's why it cannot survive mutant neurospora, right? Auxotrop. It is called auxotrop. So he understands after the mutation what happens. If some genes delete or some add, then what happens? One gene, one mutation means, he said simply, one mutation means what? One metabolic block. He gives this thesis or he gives this format, right? Now M. Swaminathan. In India, it is okay. This Wiedel-Tatum, they are foreigner, but when we consider our scientist, means as far in India, we have Swaminathan. You know Swaminathan, right? M.S. Swaminathan, right? This M.S. Swaminathan, he also induced mutation in wheat plant, right? Second here, M.S. Swaminathan, Indian, right? In India, M.S. Swaminathan induced mutation in wheat, and wheat, you know, wheat scientific name is Triticum aestium, right? In wheat, and so different varieties he obtained after mutation, right? So different varieties. Which varieties he obtained here? Sharbati, Sonora, right? Wheat, and obtained or produced, we can say, right? Different varieties. As for example here, Sharvati, Sonora, Pusa, Sharvati, Sonora, Pusa, right? This name is Pusa Lemma, etc. And he also means established Gamma Garden, right? This Swaminathan established Gamma Garden in where? IARI. You know IARI, Indian Agriculture Research Institute, New Delhi. Indian Agriculture Research Institute, New Delhi. Right? He means what? Swaminathan. Right? And this institute is called Pusa Institute. Very Pusa Institute. TITUE. Pusa Institute. This is the about of, means we can say, hey, this one is the about of some history of this mutation, right? Now we start the type of mutation, which is our main point, that type of mutation, right? When we consider the type of mutation, here we have two types of mutation. Two types means either chromosomal or it is gene mutation, right? So first, chromosomal means when mutation occurs in whole chromosome. Whole chromosome means what? Either in the number, either the number is increased or decreased first, or the chromosome structure or shape is different, right? Means it is altered. If chromosome structure or shape is altered, it is also a chromosome. It is called chromosomal aberration. And if number is altered, then it is called as ploidy, right? It is also two types. Means we can say heteroploidy, right? Another one is here gene mutation. Gene means when mutation occur not in whole chromosome but within the chromosome, means we can say it is responsible for the alteration of genes. Then it is called gene mutation, right? Here two types of mutation. Two types of mutations are there, right? Which one? First I tell you, first one is chromosomal mutation and second one is our gene mutation, right? Chromosomal mutation and gene mutation. The first one is our chromosomal mutation. Chromosomal mutation, this mutation is also two types, right? First one, this one chromosomal mutation, it is two types. Two types means first one is that is heteroploidy. Means here heteroploidy means number of chromosome altered, right? And second one is chromosomal aberration. Chromosomal aberration. That means what? Shape or size is changed. Shape or structure, we can say. Structure altered. Right? So these are two types. Chromosomal aberration. Right? This one is aberration. Chromosomal aberration. Right? When we consider the gene mutation, The gene mutation, it may be, it is at different types, right? We will read this gene mutation in another. First, we start this chromosomal mutation. The first one is our heteroploidy. Heteroploidy. Hetero means what? Hetero means different. When the number of chromosomes differ from its original, right? Differ means either it is less number or it is more in number, right? So this heteroploidy is also two types. Two types means what? Heteroploidy is also two types, right? First one is euploidy and second one is aneuploidy. Euploidy and aneuploidy. Euploidy means what? Euploidy means if the set of chromosome increase. Set of chromosome. Set of chromosome increase or decrease, right? That means it becomes N, 2N. 2N is normal actually, 3N, 4N or N itself also, or so N, right? Many extra. And aneuploidy means what? If number of chromosome increase. Number of chromosome either decrease or increase, right? So this one is trisomy, tetrasomy, pentasomy, that is called, right? Here we use haploidy, right? Haploidy, triploidy, tetraploidy or polyploidy. This side here what we use? Trisomy, tetrasomy, pentasomy, nullisomy or monosomy, right? So first we have euploidy. We read this euploidy, then aneuploidy, right? Now look here, euploidy. Euploidy means what I said you? It is the set of chromosomes increase or decrease. Because every animals means they are 2N, diploid in nature, right? So diploid is normal. If they are, okay, not all these animals, because we have some exceptions also, like male honeybee, it contains only N. N means what? It is haploid, right? Diploid is not normal. Sometimes what happens? Triploidy, tetraploidy and pentaploidy also form, right? Mostly this euploidy is present in plant, right? Okay, first euploidy. First is our euploidy. As I already tell you, set of chromosomes increase or decrease, right? Euploidy means what? Set means what? 2N, N or 3N or 4N or so N, right? So these are called, we use here haploid. N for what? Haploidy. 3N means what? Triploidy. 4N means what? Tetraploidy, right? 5 means pentaploidy, so on. Suppose that it is, how many numbers are there? Suppose that it is 6N means what? Hexaploidy. 7 means what? Heptaploidy. 8 means octaploidy, and 9 means nanoploidy, and 10 means dexaploidy. More than this one means polyploidy. If it is more, then it is called as polyploidy, right? We here what we use? Here we use haploidy, triploidy, tetraploidy. We use more in number is polyploidy, right? Okay. Now here an example is there, right? Mostly this polyploidy, if the polyploidy is present, as for example triploidy is present, so they are striped. First they are striped, right? So some examples where I tell you. Mainly understand here one thing is there. If it is in odd ploidy is there. If it is odd ploidy. Odd ploidy means what? 3N, 5N, like this, right? As for example 3N or 5N, 7N, right? Odd ploidy, it is mostly sterile, not fertile. And If it is even ploidy, it becomes, as for example, we have 4n or 6n or 8n and so on, right? So these are fertile. These are fertile. With some experiments, we can understand this one is. Do you understand this euclidy? Euclidy simply you can say the set of chromosomes is increased. As for example, in human means what? How many chromosomes are there? We know in normal human it is 46 chromosomes. 46 means what? It is 2n. But 3n case, in 3n what happens? This 46 becomes 69, and this is tri, right? 4n means what? So not a single chromosome is increased, but total 23 chromosomes, total genome is increased. This one is also called as genomatic, right? This one is also called as genomatic. Haploidy, heteroploidy, it is also called as genome, because genome is there. So genomatic heteroploidy, right? You understand? Now here you understand how it works. This heteroploidy, it is also, we can say heteroploidy means in heteroploidy, here this euploidy, or we can say polyploidy, it is also two types, right? Two types means what? It is autopolyploidy, or it is first. Auto means what? Auto means what? Auto means same set of chromosome is increased. And second one is allopolyploidy. Allo means what? It is not same, but two different sets are increased, right? Here, as for example, here, this, we can say polyploidy. Polyploidy means 3n, 4n, 5n, 6n. It is not only maximum, right? Polyploidy is of two types. Two types. First one is our autopolyploidy. Autopolyploidy means what? The repetition of same, we can say here, repetition of same set of chromosomes. Repetition of same set of chromosomes, right? As for example, in here we can write, suppose that I represent it by A. That means what? It is triploidy. Repetition of same set of chromosomes, right? The same set of chromosomes, as for example, it is present in Sanodon. Sanodon common name is doob grass. In Hindi it is called doob, right? Doob grass. And in rose, right? Because they have, I write here, these are tri, these are sterile. STRILE, right? These are sterile. Means what? Non-fertile. Means non-fertile. Second is our alloploidy. Allopolyploidy. Allopolyploidy means what? When more than one type of repetition of more than one type of chromosome set. More than one type of set is there, right? This is called allopolyploidy. As for example, we can, here I use A, here I use A, B, so on. Here two sets are there and they are repeated, right? This one is called, because they are two different sets is there, that means these organisms are also two different. They comes from different species, interspecific or intergeneric, right? Mostly here we use, they are intergeneric. They are intergeneric cross, right? They shows intergeneric cross. Generic means two different genus are used in this cross. As for example here, first example is Rapnobrassica, right? Rapnobrassica. Here you can see two different names come, Rapno and Brassica. So Raphanus sativus plus Brassica oleosera. That means here it is the cross between, look, Rapno means what? Raphanus, it is reddish, right? Raphanus sativa, cross between reddish and cabbage. The reddish name is Raphanus sativus, and cabbage means its scientific name is Brassica oleracea, right? Raphanus, we can see here, Raphanus sativus, right? This scientific name we should underline, cross with Brassica oleracea. This is the cabbage. Raphanus sativus, what is the number of chromosome here? 2x is 18. 2n, it is 18. Here it is 2n is 18. Both number of chromosome is same because both belongs to the same family, right? That is our, you know very well, this is the Crucifery. They belongs to Crucifery. Both belongs to Crucifery, right? Then what happens when we cross? We understand that n means what? 9 chromosome, and 9 chromosome from here and 9 chromosome from here, because gamete means n. Then what happens? We get second new generation. We get another 2n, that is 18, right? And this one is called Rapnobrassica. You can see here 2n means 18, here also 2n means 18. When a gamete is formed, in the gamete we know very well haploid number of chromosomes are there. So haploid number of chromosomes means only n, here only n. That means when they cross with each other, n plus n, they becomes 2n. And how many? In this case we understand that the new variety which we obtained, it is Rapnobrassica, which has 18 chromosomes. That means 2n is 18 chromosomes. But because it comes from two different genera, so it is sterile, not fertile, right? Then what happens? By using colchicine, you know colchicine is the poison of mitosis, right? Here when we use colchicine, right, then we get another means polyploidy stage. Means this, what happens when it is crossed with Rapnobrassica means selfing. After selfing, what we get here? Rapnobrassica NO Brassica, right? But this is, it becomes 4n, right? That means it has 36 number of chromosome, and this becomes fertile. This is the induced mutation. An example of induced. How can we get this? Now this is the mutant, a new variety we get, a fertile new varieties we get, right? Why we need this 4n? Because this one is sterile. Another example which is induced, which is practiced by MS Swaminathan, that is wheat, right? So we can write here another example. This is the first example. Second example is triticale. 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 Now you understand triticum aestivum, it is the scientific name of wheat, and kale, this kale, it is this one is coming from C-kale cereals. C-kale cereals means rye. So this one is the cross between wheat and rye, right? So what is the scientific name of wheat? It is our triticum, right? Durum. wild triticum durum. It is a wild variety of wheat crossed with this one, C-kale, cereals, right? That is cereals, right? Not C-R-E, it is C-E-R. C-E-R-E-A-L-E, cereal. Both are scientific name. That is rye, right? When they cross. Now look here, triticum durum. What is the this triticum durum? It is already tetraploid, means 4n is here because it contains 7 chromosomes in n. So 4x means what it is? 4n means what? 28. 7 multipliable. And this C-kale cereals, it is 2n normal. So it is 14. 2n, right? Means it is 14. You understand one thing is very important here. The number of chromosomes is n is equal in both. Then we get the new means then we are able to cross between these two plants, right? Now here you understand this one is from family, and it is not intergeneric but it is interfamiliar because this one it belongs to the family rye. Rye is the family of Cruciferae and this one is the family of Poaceae or Gramineae. So when we cross these two, we get, because here when gamete is formed, so this gamete contains 14 right chromosome from that side and this gamete contains 7 chromosome by that side. So when we cross this one, so we get in first stage we get triticale, that is triticale C-kale, right? So we here triticale, that is triticum drum and C-kale means here triti plus C, this kale. So we write here triticale. Here a new Triticale. Triticale we obtained 14 plus 7, it is 21 chromosome and 21 means what? 3n. Because it is a odd, so it is sterile. First generation is always sterile when we use intergeneric, right? Then for becoming it or convert it into fertile, from say sterile to fertile, we have to use colchicine, right? And when we use colchicine, right, then what we get? This is just double because here no meiotic division is here, right? Then what happens? It becomes 21 plus 21, 42 chromosomes are there and 42 means what? It becomes 6n, right? And this one is fertile, right? This is also triticale, but here what means we can say this is one is triticale, it is fertile. Do you understand? This 6n, this one is our triticum aestivum. Triticum aestivum, which you read in our 11th portion, right? Triticum aestivum, wheat scientifically. Okay. Now, if we suppose that, then after what happens? This Swaminathan, right? Scientist Swaminathan, sir, he once more what happens? Cross with C-kale cereals. This Triticum aestivum is once more crossed with C-kale cereals. So what happens here? We know here it is 642, right? So 6n crossed with this C-kale cereals, it is 2n. So 2n means what? 14. 6n means what? It is 42. The new generation which we get, it becomes, look here, this is 42 and this is 14. So that means 7 from this side and here how many? How many here? It is 42, half of 42, 21. So 21 from that side and 7 from that side, so it becomes 28. In first generation, 28 and 28 means what? It is 4n. You can understand here 6n means when it forms a gamete. So this gamete contains half number of chromosome. Half number of chromosome is what? It is 3n. Here also half number of chromosomes, it is n, right? Now it becomes 4n and this number of chromosomes is 28, right? Because it is the first generation, so it is sterile. So forming to means when we want to get fertile state, then what we need? We need once more selfing, right? So selfing means what? We use first colchicine and then self, right? When we use colchicine, what happens? This chromosome number becomes double. 28 becomes 28 becomes double, and 28 becomes double means what? It is 28 plus 28. It is called 56. It becomes 56. The number of chromosome becomes 56, right? 56. 56 means what? We can say by using colchicine, now it becomes 56. 28 plus now, and it is 8. 56, right? This one. It is also critical because triticum aestivum plus this one. Triticum aestivum 6n and this one is crossed with caecale clears, right? So this one is 6n and this one is 2n, right? When we cross, we get the critical and this one is fertile. Now you understand here 8n. 8n means what? It is octoploidy. Octoploidy. Example of octoploidy. So these are some examples. These are called induced, right, mutations. And these are the example of heteroploidy. Now second one is the aneuploidy. Aneuploidy. What do you mean by aneuploidy? Aneuploidy means when number of chromosome increase or decrease in a set is increased or decreased. Number of chromosome increase or decrease. There are also two types. Two types means decrease or increase. If it is decrease, then it is called hypo. First one is hypo, right? Aneuploidy. Hypoaneuploidy. And second one is our hyperaneuploidy. Hyper means addition, this means deletion. One chromosome decrease, one chromosome increase, right? So this one is decrease due to the number is decrease, and this means what? Number is increase, right? Hypoaneuploidy, hyperaneuploidy. Okay, hypo means what? Loss, and hyper means what? Gain. If it is, these are also represented by 2n minus 1. 2n minus 1 means what? Monosome. Because we know in 2n what happens? One set is there, one pair is there, right? In one pair, one is suppose that one chromosome is absent, right? As for example, we have Turner syndrome. Turner syndrome is what? In 23rd chromosome, one chromosome is absent, right? The number of total chromosome is now it becomes 45, not 46, but 45, because 23rd chromosome, 23rd pair, it is no more paired. It is only a single chromosome. That's why it is Turner. So it is called monosomy, and it is represented by 2n minus. When suppose that it is given here 2n minus 1 minus 1. 2n minus 1 minus 1. So this 2n minus 1 minus 1, it is called as double monosomy. Double monosomy. So 2n minus 1 means what? Two pairs of chromosome. Maybe 21st, 22nd, 23rd, two set. They have single. Suppose that it is 2n minus 1 minus 1 minus 1. So it is called triple monosomy, right? So this one is 2n minus 1 means double monosomy, means two different pairs they have single chromosomes. Another is your, if it is given here 2n minus 2. So 2n means what? A same single chromosome. 2n minus 2 means what? Number of chromosomes is 0, means one pair is absent. The total pair is absent. Means in 23 pair, only 22 pairs are present, one pair is absent. Because no chromosome is there, so it is called as nullisome. Right? Means one pair is absent. Okay. Same as to here also in hyperaneuploidy what happens? Same thing is here. 2n plus 1. Because here one more chromosome comes in a single pair. No, in as for example here trisomy in 21st chromosome or trisomy in Klinefelter means the 23rd chromosome. What happens here? X, X, Y is there, right? So here three chromosomes are present in a last pair, that is called trisomy. Trisomy, right? When it is given same as to here 2n plus 1 here double trisomy, right? When it is given here 2n plus 2 means it becomes 4, that is called as tetrasomy. 2n plus 3, now it is called pentasomy because number of chromosomes are more, right? So these are different terms we use for hypoaneuploidy or hyperaneuploidy, and these two types are present in aneuploidy, right? So these are, we can say, these are different type of heteroploidy. Okay.